ladies and gentlemen, where we ended off in the last screencast, we talked about the 3 different solutions you can have in regard to their water concentration. And the first one we're going to talk about specifically is isotonic. So what is an isotonic solution? And we'll see what effects an isotonic solution has on an animal cell, specifically a red blood cell. And we'll see what effects an isotonic solution has on a plant cell. And we'll do this for the hypo and hypertonic solutions as well. Keeping in mind that the reason these types of solutions are relevant to your cells is because their membranes are semi-permeable, meaning some things get in and some things get out, and they live in these water-based environments. And if the water level or concentration is incorrect, it can have some pretty drastic effects. So, isotonic is the amount of water is the same on the inside and the outside of the cell. That doesn't mean that there's no water traveling across the membrane. That just means that the water flows out of the cell at an equal rate that it flows into the cell. So the same rate, and this is indicated by our arrows here in the animal cell and plant cell picture. Now notice, in both the animal and plant cell, these cells look okay. Again, we're in a state of equilibrium, right? Via osmosis, water is diffusing out and water is diffusing in for both cells. And the cells look healthy. All is well. Now, we're going to see a different story in the other 2 solutions. First off, these are both animal and plant cells again. Red blood cell and the plant cell, they look drastically different this time, however. And notice there's only one arrow. Okay, so this is a hypertonic solution. And in a hypertonic solution, if you place a cell there, that means that there is more water on the inside of the cell rather than on the outside. And sorry about these formatting issues, folks. So there is more water inside the cell than there is outside the cell. And remember, concentration gradients. In the absence of other forces, molecules, like water molecules, will travel from an area of high to low concentrations. So here, if there's a high concentration of water in the cell and a small amount of water outside the cell, water is going to flow out of the cell via osmosis. So there's a net movement of water out of the cell. And again, sorry about these format issues, folks. So water moves out and water moves out. And notice when water moves out of the cells shrivel. They actually look dehydrated. Okay, and that's because they are. And we call this plasmolysis, which just means too much water has moved out of the cell. And this is not a term that I'm going to test you on. This is just for our learning experience here. So if I say plasmolysis, just think water moving out of the cell. Now what about hypotonic, our last solution? Hypotonic, notice here the cells look different once again, but they look the exact opposite of a hypertonic solution. That's because a hypotonic solution is the exact opposite. A hypotonic solution is if a cell is placed in this type of solution, there is more water on the outside. More water on the outside of the cell. Boy, these format issues, folks. So more water on the outside of the cell than there is on the inside of the cell. So there's more water outside than there is inside, and we follow that concentration gradient. Where's water going to move? Water is going to move into the cell, and it's going to blow it up like a balloon, basically, like you see here and here. Okay, the cells look like they swell. Now the plant cell, notice, isn't bursting like our animal cell. That's because the plant cell has this very strong cell wall that keeps it from bursting. Our cells, yours and my animal cells, don't have these. So the membrane just can't take it at some point and will break. And we call this cytolysis. Too much water moves in, and the cell membrane bursts or ruptures. And you can omit this part. We can omit this part, folks. So just skip over these 2. The turgor pressure in plant cells, not something we need to focus on here. All right, now you have these next few slides on your study guide, so I'd like to skip them. I'd like to, if we can, skip past these examples. I'd like you to do those examples in your own time, and if you have questions, please ask, but we're going to skip those for now. And the answers are in the slide set that you have made a copy of in this slide set, so you can go back and check. Now we're going to talk about facilitated diffusion. Facilitated diffusion is when you have the cell membrane, right, which we see pictured here, right, our lipid bilayer. Remember, lipids are hydrophobic, meaning they don't like water, right? Lipids don't interact with water. Hydrophobic, fear of water, right? That's what lipids are. If they liked water, we call them hydrophilic. Hydrophilic meaning they like water or they can interact. But the lipid bilayer, lipids, the macromolecule, doesn't like water. They're hydrophobic. Therefore, some things can't get through this lipid bilayer, and they need proteins like we see embedded within the membrane here. We have the outside of the cell, the inside of the cell. Some charged molecule that can't get past this lipid membrane needs to use these proteins, and these proteins facilitate or help these molecules move into the cell. And all these molecules are doing is moving from an area where you see they're highly concentrated out here to an area where they're not so concentrated. So they're just diffusing, but they're having a little help. They're getting a little help from these membrane proteins, which are facilitating the diffusion. So let's just call it facilitated diffusion. So we have polar molecules, and when I say polar, you say charged. So we know these molecules have a charge, like water and glucose. They have difficulty crossing the membrane through the lipid bilayer, which is what this blank should say, through the lipid bilayer. Again, because lipids are hydrophobic, they don't like water. So these membrane proteins or transport proteins, these are transport proteins, these purple things here, help these molecules to pass more readily through the membrane. Because your cells do need glucose, water, and the lipid bilayer doesn't want to let that stuff in, but these proteins do. So we need to get them in somehow, and facilitated diffusion is that way. So we have a high concentration outside, a low concentration of our molecules inside. We have our transport protein. So these polar molecules will pass directly through these proteins without having to come in contact with these lipids at all. And this is known ultimately as facilitated diffusion, which is a type of passive transport, right? Remember, passive transport's like this broad term, and you have things like diffusion and osmosis, and now facilitated diffusion, all under that umbrella that is passive transport, aka requiring no energy. And again, these are polar molecules, charged molecules that can't get into the cell otherwise because of our lipid bilayer, the macromolecule that doesn't interact with water because it's hydrophobic, doesn't interact with any charged molecules because it's hydrophobic. So these transport proteins help get your cells what they need. All right, folks, tune in for next time. Some active transport, the exact opposite of passive.